Hello, good day everyone. I am Lyka Bernardino, taking Bachelor of Secondary Education, major in General Science. So, our topic for today is talk about metaphase. So, what is metaphase? Metaphase is a process of nuclear division in eukaryotic cells that occurs when a parent cell divides to produce two identical daughter cells. During cell division, metaphase refers specifically to the separation of the duplicated genetic material carried in the nucleus. Metaphase is conventionally divided into 5 stages known as Prophase, Prometaphase, Metaphase, Anaphase, and Telophase. Prophase is the first stage of cell division in both mitosis and meiosis. Beginning after interphase, DNA has already been replicated when the cell enters prophase. The main occurrences in prophase are the condensation of the chromatin reticulum and the disappearance of the nucleolus. The second one is Prometaphase. Prometaphase is the second phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. During Prometaphase, the physical barrier that encloses the nucleus, called the nuclear envelope, breaks down. And the third one is Metaphase. Metaphase is the third phase of mitosis, the process that separates duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. There is an important checkpoint in the middle of mitosis, called the Metaphase checkpoint, during which the cell ensures that it is ready to divide. The fourth one is Anaphase. Anaphase is the fourth phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. The separated chromosomes are then pulled by the spindle to opposite poles of the cell. And the last is Telophase. Telophase is the fifth and final phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Telophase begins once the replicated paired chromosomes have been separated and pulled to opposite sides or poles of the cell. So let's move on. What is meiosis? Meiosis is a special type of cell division of germ cells and sexually reproducing organisms used to produce the gametes, such as sperm or egg cells. It involves 2 rounds of division that ultimately result in 4 cells with only 1 copy of each chromosomes. There are 6 stages within each of divisions, namely Prophase, Prometaphase, Metaphase, Anaphase, Telophase, and cytokinesis. In this article, we'll look at the stages of meiosis and consider its significance in disease. Prophase 1. Prior to prophase, chromosomes replicate to form sister chromatids. Prometaphase 1. Spindle fibers attach to the chromosomes at the point along the chromosomes called centromeres. While this is happening, the chromosomes continue to condense. Metaphase 1. Maternal and paternal version of the same chromosome align along equator of the cell. Anaphase 1 begins when homologous chromosomes separate, the nuclear envelope reforms and nucleoli repair, the chromosomes collapse. the nuclear membrane begins to disintegrate and the centrosomes begin moving apart. Spindle fibers form and sister chromatids align to the equator of the cell. Telophase and cytokinesis 1. During telophase 1, the nuclear envelope reforms and spindle fibers disappear. In cytokinesis 1, the cytoplasm and cell divides resulting into cells are technically haploid. There is one chromosome and two chromatids for each chromosome. Here each of the homologous chromosomes get pulled toward opposite poles of the cell as spindle fibers retract. This equally divides the DNA between the two cells which will be formed. Meiosis 2, Prophase 2, and Prometaphase 2. These stages are identical to their counterparts and Meiosis 1, Metaphase 2. In Metaphase 2, chromosomes line up in single file along the equator of the cell. This is in contrast to Metaphase 1, where chromosomes line up in homologous spheres. Telophase 2. This stage is the same as Telophase 1. Cytokinesis 2. Again, the cytoplasm and cell divides producing two non-identical haploid daughter cells. As this is happening in both cells produced by Meiosis 1, the net product is four non-identical haploid daughter cells, each containing one chromosome, consisting of one chromatid. These are polyform gametes. Anaphase 2, next sister chromatids are pulled to opposite poles of the equator.